Ladies and gentlemen, welcome to this latest teaching session in the course on Technology and the Future of Medicine. Today we have Quantum Biology Part 2, also known as the mystery of the human brain, memory, information processing, neurodegenerative diseases of the subcellular quantum level by Jack Tuszynski. Jack, take it away.
Thank you, Kim. I've decided to spice it up a little bit. So you may recall that last time I set the stage for the quantum revolution in biology. Unfortunately, there's not a heck of a lot of hard evidence for quantum effects in biology. There is growing evidence, and there is, of course, a lot of evidence that classical mechanics doesn't explain a lot of things. And I think the most of, in terms of biology, most of the puzzles come from neuroscience. And today I'll try to portray a little bit of the enigmas and maybe offer some solutions to some of the key problems in neuroscience which involve memory, information processing, and consciousness. Big topics, deserving probably a full semester course rather than one lecture. If you want to find out more about this, I co-edited and co-wrote 2 books on these topics. One is called Nano-Neuroscience, which really deals with sort of technological biotech aspects, how we can harness the power of nanotechnology in the context of neuroscience. And the other is more esoteric, the emerging physics of consciousness, how quantum mechanics in particular and other physical concepts can help us understand consciousness, the biggest enigma of them all. So I'll just review very briefly where we ended on Tuesday. There's a little bit of, well, maybe more than a little bit, evidence for quantum coherence at the level of photosynthesis, and that's been accumulating over the last decade and actually being published in some very reputable journals, and both experiments and theory are consistent in these explanations. So it has to do with light absorption at the quantum level by both plants in terms of chlorophyll and the photosynthetic processes that follow, and in some bacteria which are thermophilic and they utilize light as well for their energetic needs. This is the molecular structure of this so-called FMO complex in these bacteria, C. Tepidum. It's just a nice painting, but you see a regular arrangement of heterocyclic molecules, and in terms of biochemistry, tryptophan is one of those amino acids that is very susceptible to light absorption and emission, and we'll come back to it in a few minutes because there's so far theoretical but rather convincing evidence that it goes beyond bacteria and plants. It may also take place in mammals and in fact in the human brain through tubulin microtubules, which play a very important role. I'll say something about that in a few minutes. But I just want to draw your attention to the structure. The structure in biology is all important. Structure leads to function. And if you see similar structures, usually you find similar functions. At the end of last lecture, I mentioned to you that what differs between living and non-living systems is energy transduction. And living systems exist at non-equilibrium or far from equilibrium conditions. They must periodically receive injections of energy in the form of nutrients, and that eventually leads to the formation of ATP and GTP molecules that are the common currency for all metabolic processes. And these are examples of just the 3 major types of structures that transduce energy. Chloroplasts in plants, as you can see on the right. Bacterial membranes with various biochemical complexes, and most importantly, mitochondria in higher organisms. Mitochondria actually are organelles in eukaryotic cells that are very complicated, and they have actually highly complex organization of protein complexes in the walls. There's 5 complexes, each of which is highly structured, and importantly, it has electron hopping mechanisms that drive the proton pumps and eventually ATPases, enzymes that release ATP into the cytoplasm after a number of steps. I don't remember exactly, 7, 8 steps, something like this. But the most important thing is that the mitochondrial walls, the 5 complexes, especially complex 1, have reactive centers which are close enough for quantum tunneling to take place. You remember tunneling? We talked about ghosts and so on. That's really limited to some extent, to our knowledge, to distances more or less under 20 angstroms, 2 nanometers. Beyond that, this tunneling at the level of electrons does not occur. There's not enough energy to tunnel electrons in solid-state physics, let's say. In biology, maybe more, but nonetheless, we have this condition satisfied in complex 1 in mitochondria. There has been very little research on this, but it's happening. So what I want to say is that my next best candidate for quantum processes in biology would be mitochondrial energy transduction in complex 1, and be on the lookout for this. But we do have indirect evidence. So far, I'm pointing at direct evidence for the emergence of quantum processes in biology. Indirect evidence actually is quite interesting. And you may recall from last lecture, I historically painted a picture, a metaphor between physics and biology. And in physics, there were a lot of experiments that all documented the prevalent paradigm of quantum mechanics at the level of elementary particles. 2 most important that I indicated were the photoelectric effect and the black body radiation effect. The third one that I think I mentioned in passing, maybe I didn't have enough time, was specific heat of solids, which Einstein tried to explain and failed. Paul Debye did and got a Nobel Prize, of course. And there is something very similar in biology to the specific heat of solids, namely metabolic scaling laws or allometric scaling laws. I don't know if you've heard about this or not. It's actually a somewhat arcane topic, but it recently rose to prominence because it is so pervasive throughout various kingdoms. Basically, what it says is the following: the power consumption of any organism, ranging from a bacterium to an elephant, scales according to its mass to this power law. P is power, W is the weight of the biological system, organism. As I say, it covers all of life. And the exponent beta is in a very narrow range from 0.5 to 1. Actually, most of the examples of this scaling law involve very specific value of beta, namely 3/4 in animals. All animals satisfy this power law. In other words, if you were to, if you had a cat and then suddenly decided to have a dog, and the dog would be 10 times or 8 times the mass of the cat, you'd like to figure out how much food to give to the dog, you would probably automatically think that it's 8 times, right? Because it's 8 times bigger, it'll have to have a demand 8 times greater. It's not true. It's actually 8 to the power 3/4. So whatever that is, I can't do it in my head, but it's probably something like 6 or close to it, not 8. So, and there is no explanation for this, actually. There was a lot of discussion in the literature in the late 90s and early 2000s about this. Some prominent people in the field proposed that this is due to the fractal vasculature, delivery of blood, and blood delivers nutrients to all cells, and it has a very pronounced branching effect, which is called fractality. But of course, bacteria don't have vasculature, and lower organisms. So that may be true for mammals, but doesn't explain the whole range of examples throughout biology. Single-cell organisms and very primitive organisms, they satisfy the same law. So actually, this law was proposed by Kleiber following the work of the intellectual giants of 18th century, Lavoisier and Laplace, and then Rubner in 19th century, and then Kleiber in France eventually classified it, codified it, and came up with this simple formula. My colleague Lloyd Demetrius from Harvard came up with a very simple explanation based on a formal mathematical analogy with specific heat of solids. Namely, it involves enzymatic, all of these energy-producing reactions involve enzymes and actual enzymatic oscillators, cyclic reactions taking place in living systems. And if you assume that these are oscillators, remember in quantum mechanics we talked a lot about oscillators. The harmonic oscillator is the blueprint for all things quantum. That's the first example of quantization. You know, what people like Schrödinger and Heisenberg solved were the harmonic oscillators. So if you assume that biological oscillators are also in some way quantized, and I tried to convince you that energy is quantized in biology by the units of ATP or GTP, 10 to the minus 20th joule, maybe you can accept this. Actually, there is now recent evidence to back it up from NIH. Actually, this year there was a paper published showing that in tissue, metabolic enzymes synchronize. They actually oscillate at the same frequency, although cells they have their own autonomy. But when you put it in a healthy tissue, there is synchronization, and these reactions take place simultaneously, in sync. Why is this important? Because I think biological organization, as I mentioned earlier, is incredibly efficient. And just like efficient societies maybe use fewer resources and accomplish more with the same amount or even less because of perfect organization and sharing of resources and proper timing, no delays and so on. The same applies, in my opinion, to tissues and organs of the human body or animal bodies, where cells are not acting individually. They act as members of a society of cells for a particular organ or tissue. Where this breaks down, by the way, is in cancer, where each cell acts on its own accord and for its own benefit, and that leads to major problems. In fact, this includes also metabolic demands. But I have no time to go into it. It's a separate topic, incredibly interesting. If you're interested in cancer and metabolism, you should read the work of Otto von Warburg, a Nobel Prize winner from Germany in the 1920s. He, for the first time, proposed this metabolic origin of cancer. But anyway, back to the quantization principle. So in normal tissue, let's forget about cancer for now, enzymatic oscillators synchronize, and so Dr. Demetrius proposed, you have the reference here, now more than a decade ago, that you can quantize it just the way Planck quantized electromagnetic radiation. This is now metabolic oscillators, not electromagnetic oscillators. If you just make this assumption, and they make a formal analogy between solids and cells, so physical systems and biological systems, where temperature, because in thermodynamics, of course, temperature is the independent parameter, but it's replaced by something else in biology, cycle time. These are dynamical systems. Heat capacity or specific heat of solids is replaced by the metabolic rate, P. Remember the metabolic rate P in this formula, allometric scaling law. And the thermodynamic entropy is entropy production rate. Or entropy production rate is really the rate of heat production. So if you make these formal analogies and do some simple math, a few integrals, which are actually standard in solid-state physics. This integral at the top earned Paul Debye a Nobel Prize, by the way, because it really explains how specific heats depend on temperature in to power 3 cubic power of temperature, absolute temperature, for solids, which is well known from 1930s or so. You end up with, so I'm not asking you to solve this integral in your head, because it also requires some clever approximations and taking some limits for high and low frequencies. But the bottom line is, actually at the bottom, that for relatively fast cycle times for these metabolic enzymes, you end up with a scaling law where you have the P, the power production or consumption of a living system, scales with the mass, or weight, W, to the power D over D plus 1. D is the spatial dimension. So if you had a flat organism, or metabolic enzymes would more or less span a plane, then this would be 2/3. The scaling law would be between the power consumption and the weight to power to 2/3. If it's three-dimensional, which is most of the time, you may have some flat organisms or culture of cells or bacteria more or less flat, then I grant you that. But most of the time you have D equals 3, three-dimensional distribution of oscillators, you have 3/4. So you exactly obtain Kleiber's scaling law by assuming that metabolic enzymes that produce the biological energy or biochemical energy are synchronized and distributed through a three-dimensional space. That's it. So this is what I call an indirect evidence for quantum effects for biology. Why is this important? Why? I mean, there's nothing more important than metabolism to any living organism, right? The first principle of living systems is to be alive, and you cannot stay alive without nutrients and energy production. So this is this formula. You can go into details if you're interested. The work of Dr. Demetrius is the place to go. It's not too difficult to read. Okay, that was just basically the end of the first lecture. I wanted to link it to what I'll be talking about today. Today we will be switching gears a bit and move towards the issues of neuroscience, brain, as actually we ended up with a question. I asked you, what in your opinion is the most mysterious fact in biology? I think the human brain, how it works, how powerful it is with such a small amount of mass and space and energy consumption. So we'll try to understand what's going on from the point of view of potentially quantum effects, definitely molecular level. So these are the big questions about the brain. What makes the brain special? Well, what is consciousness, which most people agree is the product of brain functioning. Some people believe that consciousness exists without the brain, but I'm a scientist. Okay, we'll not go into the religion today. Where is memory stored? What is memory, by the way? And what is the computational power of the brain? There is actually there's a long history of debate how much memory is stored in the brain and how fast the brain can function, how many bits of information can process and store and retrieve. So now to the technical issue, is information processing in the brain classical, quantum, or maybe fractal, resonant in some way, or maybe it's a hybrid combination thereof? That's my opinion. Or something else altogether. And finally, how can the brain work so well with so little power consumption compared to computers? I indicated last time a little bit about this. So, to a physicist or a computer scientist, the brain is just a large computer, actually a small computer, but very powerful. So, it has 10 to the 11th neurons, 100 billion neurons on average, give or take. the weight is between 1.2 and 1.5 kilograms. You have that many neurons and other cells as well, by the way, glia and astrocytes and so on. But neurons are the sort of functional elements. The other cells are support elements. Now, on average, a neuron will have 10,000 synaptic connections, communications with other neighboring neurons, up to 100,000 in some cases. One neuron can communicate with 100,000 other neurons. So I sometimes compare this to the world. You know, we are not there yet. The world has, what, 7.2 billion people. We have 100 billion neurons, so it's like 10 planets. But each neuron talks to at least 10,000 other neurons and sometimes 100,000. I guess if you're a celebrity, maybe you tweet to 100,000 people, or maybe 1 million, so we are getting there as well. But if you're an ordinary person like me, I probably connect with 100 people at most. Okay, so this is that. We have 10 to the 16 synapses. So synapses are the connections where the electrical activity takes place, excitation, inhibition, and the synchrony throughout the brain. Now, I'm going to make a comparison to this Blue Gene supercomputer that we actually acquired in the fall for $2 million. It's one of the best supercomputers in the world, one of the most powerful. It has a performance of 10 to the 15 flops, so 1 petaflop. Approximately. That means 10 to the 15th. In human language, it's 1 million billion operations a second. That's pretty fast, right? But still, if one synapse operates, has 1 operation a second, which is really a very low, almost absurdly offensive estimate for the function of the brain, we are beating the supercomputer on the number of operations per second in the brain, hands down. Now, the total consumption of the brain in terms of energy is 25 watts, as I mentioned to you. It's about 25% of the human demand, the entire body's demand on energy. By the way, 70% of this goes towards maintaining temperature and protein synthesis, maybe even more than 70%. Very little is used for operational capacity, just running the processing. Maybe 3 watts, I would estimate, perhaps less. Compare it to the Blue Gene's requirement of 1.5 megawatts of power consumption, with probably a lower performance in terms of number of operations. So my claim is that the human brain is a supercomputer, perhaps even more than that. And each neuron is, to me, already a computer, a very capable processing unit. So the question is, if the brain is more than a computer, and the neuron, each cell, is already a computer, not a supercomputer, perhaps, what's inside the neuron, and what's happening there? Is it just, bursts of currents going through the brain, and that's it? Well, actually, the structure of the neuron is quite complex, and most of the people don't look inside. They simply look at the connections and measure the currents across the membrane, action potentials. But so you have the axon, the long arm of the neuron, and you have dendrites, where most of the interactions with other neurons take place through dendrites. And inside the neuron, the so-called soma, the body of the neuron, you have the nucleus, and then these segments, bar segments in solid black, are actual microtubules, interconnected by little short segments called microtubule-associated proteins. So this is a network of protein filaments, densely packing this structure of the neuron. Roughly speaking, each microtubule inside the neuron is separated by about 90 to 100 nanometers of space and connected. This is an architecture of great complexity. Microtubules MT run along the dendrites. And when you have the synaptic connection, these other little filaments are called actin filaments or microfilaments, a different type of protein, but they are also interacting with each other. So this is not done in isolation. What's important is that the brain, we keep hearing that the adult brain, we don't grow any new neurons once we become adults or even earlier. But it doesn't mean that the human brain is static and doesn't change over the lifetime. We actually may not grow new neurons, but we could grow new connections or lose connections depending on the usage of the brain. So it's called neuronal plasticity. The neurons which are being used a lot and connect to others, they get strengthened interactions. They get more and more stronger connections. This shows you that microtubules, when you look more closely, microtubules not only form these bundles, but they also affect the functioning of ion channels. You see ion channels in the membrane, voltage-gated ion channels. They close and open and allow the flow of ions in and out of the neuron. And they are being affected by microtubules. So there is quite a bit of interplay. We don't know exactly how it's done, but it happens. Our challenge to understand the functioning of the neuron as not just as a binary unit, 0-1 switch, which is more or less the current paradigm. I think it's vastly offensive to the complexity of the neurobiology of the nervous system. So you have to look inside. When this cartoon shows you a structure of a bundle of microtubules with actin filaments going into the synaptic spine and interconnected by microtubule-associated proteins or MAPs. There's, by the way, and I'm not going to get into it, but there is a veritable beehive of activity inside. This is not just sitting there and doing some electrical processing. There's actual transport through motor proteins, kinesin and dynein, propagating through microtubules, and actin also is involved in transport. Yeah, it's very dynamic. You see, you go inside, and we'll look more closely at what's the composition of microtubules in terms of the building blocks, tubulin dimers, alpha, beta, and this protein is ubiquitous in all living systems. Actually, eukaryotic cells, without an exception, produce tubulin. Prokaryotic cells produce something which is analogous to tubulin called FTSZ or Z. So we have microtubules composed of tubulin, very fascinating structures, incredibly symmetric, incredibly diverse, and self-assembling, which is also interesting because if microtubules perform any computational functions, then maybe we can mimic this technologically and make hybrid devices in the future, maybe even memory augmentation or some sort of brain augmentation by improving on Mother Nature's design. So I guess this is the pitch towards transhumanism to really something far out, where we try to understand how biological systems work and improve or maybe augment them with additional devices or replacement of organs, and maybe even for traumatic brain injury, where, by the way, in traumatic brain injury, there's ample evidence microtubules break up into pieces inside the brain. You've heard a lot about athletes having concussions, and that results in brain damage, sometimes early demise of these players, top athletes, hockey players, baseballs, and so on, and football players. And when they have concussions, microtubules in the brain get shattered into pieces, and it's very hard to reconstitute that. You can't read this, but I'm sorry. I simply want to say that it's a complex story because tubulin, which makes microtubules, interacts with at least 200 other proteins. I mentioned a few, microtubule-associated proteins, MAP2, MAP-tau, and so on, MAP1, the subclasses and subtypes. Why am I mentioning this? Because in addition to traumatic brain injury, again, without an exception to my knowledge, every neurodegenerative disease that we know of, be it Parkinson's, Alzheimer's, Lou Gehrig's, you name it, even epilepsy, is related, if it's not in a primary way, then secondary to some degeneration of the cytoskeleton. The neurons have defects in either microtubules themselves or microtubule-associated proteins, either structural or functional. Maybe traffic is impeded. So I mentioned to you kinesin and dynein traffic. They move RNA and other material to the synaptic connections, which is used for the assembly of neurotransmitters. So if this traffic is impeded, or the pathways through which material moves are damaged, then you will have cognitive impairment or any sort of motor diseases and things like that. So in the future, we may be, I'm sorry, the red is just not very easy to see. In the future, we may be able to bioengineer repairment of microtubules in the brain by maybe genetic engineering or some other form of delivery. So, okay, this is supposed to explain what's happening in Alzheimer's disease. I'm giving you sort of a lengthy introduction into neuroscience through pathology because it's our indirect way of analyzing what's happening inside when something goes wrong. Alzheimer's is a very horrible disease and more prevalent in our society due to longevity, among other things, but has two major molecular explanations. Two competing camps of scientists, but probably the explanation is somewhere in the middle. Both things happen. The first one is tauopathy. What that means is actually in Alzheimer patients' brains, MAP-tau, this little black segment I showed you that interconnects microtubules, does not stick. It's hyperphosphorylated, and it falls off microtubules and forms clumps. And that weakens microtubules, which in the end also break down and then leads to the death of neurons. That's one camp. The other camp puts emphasis on beta amyloid plaques outside neurons. There is a protein called beta amyloid, which actually is also dysregulated in a host of diseases, including mad cow disease, through the prion accumulation. But there is a connection. Beta amyloid has a very high affinity for zinc. Microtubules require zinc for assembly. In the brain, as in all the cells, microtubules are regenerated periodically in the brain very slowly on the time scale of maybe weeks. In other cells, of course, on the time scale of cell division, which is hours, several hours. But neurons don't divide, but they still have to replenish new tubulin into microtubules. So this process requires zinc. If you don't have ample supply of zinc, will not proceed or will be very slow, and therefore microtubules will not regenerate, and then you have the consequences that I outlined before. So there is a connection. In other diseases like Parkinson's and Huntington's, there are some other culprits, like in Parkinson's alpha-synuclein, but there's also well-observed and reported deterioration of the cytoskeleton and motor transport. I'll just skip this. And I hope I convinced you that it's worth understanding tubulin and microtubules vis-a-vis the brain and neurons, because if something goes wrong, usually microtubules and tubulin are involved. Luckily, we understand tubulin very well these days. Tubulin was crystallized in 1997, and many times after, we have a lot of crystal structures of tubulin. We can construct very accurate atomistic models of the protein, and also we can model protein-protein interactions as well as protein-ligand interactions. We can find out how to repair tubulin with different pharmacological agents. So I would say the following. This has not happened yet, but in terms of advances in neuroscience, we are just turning a big corner. I think cancer has, of course, occupied the army of medical researchers for decades, and who used up billions of dollars of funding. And there is progress, of course, but it was because of the understanding and ability to do research on cells and on animals. For neurodegenerative diseases, it's very difficult to do the same. But now that we have insight into molecular structures of these players, we can do that as well. We can do a lot of things in silico. We've reconstructed. You see a cross-section of a microtubule in the top left corner. So it's a hollow cylinder, about 15 nanometers in diameter inside, 25 outside. On the right, you have this axial projection. It's a very regular structure. It's not only very regular, but it's also very stiff, very rigid compared to other biological systems, because it provides support and also pathways for transport. So it has to be straight, regular. It's perfectly symmetric, actually. I want to draw your attention to these little tails, protrusions called C-termini. Every protein has a C-terminus and an N-terminus. For microtubules, C-termini form a very beautiful structure. A colleague of mine, Dan Sackett, called it the toilet brush model of a microtubule. They actually are not just for decoration. Most of the protein-protein interactions involving tubulin takes place on C-termini. C-termini interact with kinesin, with dynein, with MAPs, and so on. So, and by the way, tubulin is a protein, but actually it's a family of proteins, encoded by about 20 genes in the human body. Different variants of tubulin exist, different cells express it differently, and it's also an address. So each tubulin has its own address label. C-termini are completely unique. You can recognize which tubulin it is by identifying the sequence of amino acids in the C-terminus. So it's very convenient if you want to find out which tubulin is where. You can simply find antibodies for each C-terminus and label it this way. Okay, these C-termini actually are very dynamic. So it's a dancing toilet brush where all the whiskers move around in a dance. Maybe you can see some of that. I've assembled here, well, actually only one, no, two are moving now. This is from simulations. Each of these is a different address label for a different tubulin, different C-terminus. All of them have different modes of oscillation. This, by the way, since we're talking about quantum effects, may be incredibly susceptible to electromagnetic radiation, and can be stimulated by it, because that's where 40% of the electrostatic charge is located on tubulin. About 10 net charges reside on each C-terminus. So moving charges produce currents and electromagnetic fields. It's almost like a radio transmitter. You can imagine that whether it's used by the cells or not, it's irrelevant right now, but it could be used for signaling at a distance. Inside tubulin, there is an electrostatic potential difference between the inside and outside. Inside also there is a potential well, which was hypothesized to allow electronic transitions, hopping of electrons between two positions separated by about 15 angstroms. Tunneling, again, a quantum effect. So if there is tunneling in neurons, it will probably involve electrons tunneling inside the structure of the tubulin. So this goes back actually to a very old hypothesis by Stuart Hameroff in 1980s that higher cognitive functions, in particular consciousness, are related to electronic states of proteins in the brain. And if anything can do this, tubulin is the place. This is the structure of tubulin with a dipole flip, and flipping of the dipole would be basically an elementary unit of information processing. This can easily go into quantum realm, and in quantum computation, you may or may not know, but bits in classical computers are replaced by qubits. Bit is a 0 or 1, right? You flip the state from 0 to 1. It's either conductive or non-conductive. But remember, in quantum mechanics, we have the possibility of being in both states at the same time. Why is this important? Because a bit has only, in terms of the information content, is telling you only one thing. It's either up or down. It's either 0 or 1. A qubit has actually a huge information capability, because suddenly from a single number, 0 or 1, you go into a complex number because the wave function is actually alpha 0 plus beta 1. It's a combination, superposition, where alpha and beta are complex numbers on the radius 1 in the complex plane. So it's actually an infinite number of possibilities. The phase, you can think about it as the phase. Okay, the phase is 360 degrees. And you think about a unit vector, if you remember first year algebra or whatever. So the question is, where is this unit vector pointing, the direction? And you have 360 degrees to choose from. So that's basically the amount of information that can be stored in a qubit using very simple, oversimplified language. Okay, so if this is a quantum tunneling phenomenon taking place on each dimer of tubulin, there's 13 protofilaments in microtubules, and a 10 micron long microtubule will have about 1,500 dimers. So each dimer is a qubit. Now you can think about the amount of information that can be stored. Huge. Okay, we'll come back to this in a few minutes. What is memory and where can it be stored? Do you have any ideas about human memory or animal memory? What is memory? Well, nobody really knows, so don't be shy. Memory is basically ability to encode, store, and recall information, right? If you walk into this lecture hall, you don't have to consult the map. You memorize the route, or you have some landmarks and you orient yourself by it. It's somewhere in your brain, right? Where is this? Well, that's the big question. So if we take the brain as a huge number of neurons interconnected by synapses, maybe it's stored in some of these synaptic connections. But it's probably more like a dynamic thing. If you reinforce the memory by recall, let's say you come here every day to the same place, you reinforce the memory, the synaptic connection gets stronger, and you need a weaker signal to activate it. For example, you don't have to look around very carefully or compare to the map to find your way. You simply notice one building or maybe a tree, and that's enough for you to find your way. So it's called long-term potentiation in neuroscience. The question is it achieved at the level of molecules, proteins? There's various theories about this, actually going on for a long time. some people claim that memory is holographic. We have a holographic image in the brain, three-dimensional image somewhere, where we kind of reflect our visual input into some sort of representation in the human brain. More recently, Anirban Bandyopadhyay proposed actually that memory is fractal. It actually has a structure within a structure within a structure. We may have short-term memories, long-term memories within short-term memories within long-term memories, and maybe several levels of this. People like Rupert Sheldrake, a very controversial biologist, claim that actually memory is not stored in the brain at all. It's a field that we access somehow by the function of the brain, but it's not physically in the brain. And of course, there's also the question of primitive organisms like bacteria, ants, and swarms, slime molds, and so on, where you have some sort of a collective memory. Individual agents or individual ants, an individual ant may not have a memory, but a society of ants may communicate collectively and store memory of the landscape, of the directions, and so on, as a colony. However, we know that memory has to do with synaptic connections and neurotransmitter release. These are two synapses, and that is related actually to calcium currents across the synapse and into the dendrite. So calcium ion is very important in activation of neuronal processes, has also an effect on microtubules. Actually, calcium causes disassembly of microtubules, but has other effects as well. So this is a micrograph of a microtubule forest, if you will, with these map connections inside the dendrite. And it's known that, as I mentioned to you, diseases cause, or maybe are caused by cytoskeleton dysfunction or damage, but conversely, learning strengthens cytoskeleton, microtubules. Now the question is, what is our capacity? How much can we remember in terms of information, bits of information? So there is some disagreement, big disagreement. John von Neumann, who is the undisputed father of computers, in 1950 claimed that the total human life experience is 3 times 10 to the 20th bits. You can't read it, that's in red, I'm sorry. 3 times 10 to the 20th. That's according to John von Neumann, one of the smartest people who lived in the 20th century. And then people were less optimistic. Anatomists in the 1970s claimed that our memory is more or less comparable to the number of synapses, 10 to the 13th, maybe even less, 10 to the 15th. If we have 10 operations per synapse a second, then you have 10 to the 16th operations per brain. Thomas Landauer wrote a paper in 1986 with a very pessimistic scenario that only 10 to the 9th, 1 billion bits of information, is stored in our human brain. I think it's really insulting to the human intelligence. He calculated it very simply. He said the human lifespan, 70-some or 80 years, whatever it is now, counted in seconds is 2.5 billion seconds. And we can store about 2 bits of information per second, so you have 5 billion bits per lifetime, if you can recall it all. I think it's very superficial, but I don't know, maybe you think differently. So there were some recent experiments about 5 years ago done at NIH in the US where mice were trained to solve some mazes, and then they were subjected to pharmacological intervention, which actually inhibits a particular enzyme called calmodulin kinase 2. And it turned out that they forgot. When they were under the influence of the pharmacological agent, the drug that inhibits the enzyme, they didn't remember the path. Okay, so that seems to have indicated a particular enzyme, molecule in memory formation or memory maintenance. This shows actually this enzyme's calmodulin kinase 2 is activated by calcium. Remember I told you about calcium waves? And it's shown here in this kind of hexagonal structure, like a flower at the top. I'll give you better pictures of this in a second. And it's very interesting, to me at least, this enzyme activated by calcium is formed, it's a complex, and it phosphorylates microtubules. Now you inhibit this, you don't phosphorylate microtubules. Phosphorylation is a major signaling phenomenon in biology, and memory is gone. So together with my former student and a collaborator, we looked at what is actually calmodulin kinase like structurally, and what does it do, and what can it interact with on microtubules. So this is actually a computer rendition of this enzyme in different views. You see, it looks a bit like a snowflake, maybe, horizontally. Vertically, it's very complex. A friend of mine called it the nano poodle because it's actually nano-dimensional. It looks like a poodle, the dog, you know, French poodle. But it has 6 legs, so I don't know, transformer, whatever. So this enzyme is a motor enzyme. It walks on the microtubule. And we reconstructed this computer image and then overlaid it on the computer-generated image of a microtubule. And it actually fits beautifully. I apologize again for this. The slides don't come across very colorfully. They are very colorful and pretty. Okay, here is the visual rendition of calmodulin kinase 2 and the lattice of tubulin on the microtubule. And then on the right you have the overlay of this enzyme. So it fits into a patch of 7 tubulin dimers, and it can phosphorylate up to 6 of them, of the dimers. 0, 1, 2, 3, 4, 5, 6, 1 in one event. That shows you a different image from a different angle. Now you see this enzyme sitting on the surface of microtubule in different, and the whiskers, the lines, show the attraction, physical attraction. There is physical forces of attraction, electrostatic forces. Each phosphorylation event costs 1 ATP, 1 quantum of energy. So you can encode at least 1 bit per phosphorylation, but you have up to 6 places where you can do it simultaneously. And this is a more dramatic representation of this event. So what I'm trying to say now is that we proposed in this work that there's a molecular interpretation of memory formation by phosphorylation through calmodulin kinase 2 phosphorylation of microtubules, and you'll see in a moment we call it a memory code. You can store a huge amount of information by doing this. You can write it and read it. This is the information content, you know, zeros and ones in the patches. And you can read it, but basically each CAMK2 may encrypt up to 5,218 bits. So that's quite remarkable. And when you calculate it per brain, it becomes now comparable to what John von Neumann said, 10 to the 20th or so bits per brain just by phosphorylation events. Okay, and this is just different states. Each state may have some information. But of course, you want to know functional what it means. As I said, the easiest way would be to interpret it as traffic lights. And this can affect neural function by traffic, as I said, the traffic of motor proteins, or, that is supposed to be microtubules interconnected by maps. So these connections between microtubules are dynamic as well, strengthening and weakening depending on where the maps go. And these are the potential AND gates on the left and exclusive OR gates. If you have more than one map nearby, then ion flows can either cancel each other or they may enforce each other depending on the rules. So that is speculative, I admit. Experiments need to be done on this, and hopefully will be done. But it gives us some idea. This was published in 2012, generated actually quite a bit of international attention, because that's the first, in my opinion, that's the first molecular model of memory at the level of inside neurons, not some field or something, I don't know, or something esoteric. This is very concrete, it's testable. So now let's put some numbers together and see where we can go with this. So potential for memory storage and computation in the brain using microtubules is enormous. I showed you one example. There are some other possibilities with C-termini. They also have at least two states in which they exist, bound to the surface and away from the surface. Again, information is always for something, right? It's instruction, really. Information is readable to somebody who understands the code. So if you don't speak Chinese, as I don't, to me that has zero information content. For somebody who speaks Chinese, there could be a poem. So you need to know how to read this encoded information. So for C-termini, this could be, again, trafficking or electric signals or some sort of amplification. You may program the neuron for sensitivity to interactions with other neurons. So anyway, if you take into account all these things that tubulin can be encoded for, there may be up to 128 bits of information per dimer, and then I think there's 10 million tubulin dimers per neuron. It's 10,000, I think, microtubules, and maybe a few thousand microtubules per neuron, and each will have a few thousand dimers. So if you do the multiplication, you end up with 1 gigabyte of information storage per neuron. So it's a memory stick. Our memory stick is about, now it's maybe more, but it used to be a gigabyte or 5 gigabytes. I don't know what it is now. Memory sticks for the computer. So think about a single neuron, single cell having this capacity for memory. So suddenly it's not so stupid. It's not a 0-1. It has very rich, vast amount of information that can be stored and utilized, because the cell needs to respond to signals from the outside. It moves, it takes nutrients and sends signals out, and so on and so forth. So we have 100 billion neurons. See, if you do the multiplication, you get 10 to the 20th bits per brain. Remember what John von Neumann said? 3 times 10 to the 20th. We're in the same ballpark. Of course, in 1950, we didn't know even that DNA existed, let alone microtubules in the brain and all this architecture, not to mention the dynamics. However, okay, so that's how much you can store. The question is how fast you can change information. This is processing, right? Storage is one thing. You can keep it there and retrieve it next year. That doesn't cost any energy. It can be lost, degraded by thermal noise, right? Noise degrades information, as you know. You may talk to somebody on the phone, but reception is bad and you lose some words. But if you process information, the question is how fast you can do it. Yes?
When you say that the information is stored in the tubulin dimer as a bit, so you say like in 6 you can have, that, if I understand well, is that is the phosphorylation state that is giving the value of the bit, the state of the bit in each
Well,
tubulin?
each tubulin dimer has many physical states that it can be in, including being phosphorylated, and there are several places it can be phosphorylated. It can have C termini away, or so this is a single state space for a single dimer. And then, of course, you have many, many of these. So I'm calculating the collective state of the brain.
Okay, so it's not just 0 and 1, so phosphorylated, not phosphorylated. It can be like from 0 to, I don't know, what was 8, 9, I don't know, maybe more, I don't know. It can be like the C terminal in some state, then phosphorylated.
Sure, yes,
like this.
yes, exactly. It could be a product of these things, direct product.
Yeah, okay.
Right. So, anyway, back to the numbers. And now I'm looping back to energy. Information, we all are in love with information in this day and age, but physics is kind of ignorant about information, but it's very competent about energy. So information transfer, believe it or not, costs energy. And somebody, also by the name of Landauer, but not this Landauer, not Thomas, Rolf Landauer, calculated the minimum amount of energy that is required to change one bit. What's the absolute minimum you require? And actually, it's kT, Boltzmann constant times the absolute temperature, times logarithm of 2. That's the theoretical limit. You cannot buy information more cheaply than this. It's called the Landauer limit, which is a few times less than the hydrogen bond. Hydrogen bond actually is a very efficient way of encoding information. It's almost the cheapest way of doing it. So, the question is, remember what I told you in the beginning, that the brain is incredibly efficient? It only utilizes about 3 watts of energy for information processing, right? 3, it's nothing. So how many bits can you change with this amount of power supply? So that depends on the speed of information change. If you have 10 to the 20th, and the so let's see. I don't think it's explained on this slide. I don't remember all the numbers. Okay. So yes. Yeah. Actual biological energy cost is much higher. The ATP. Remember ATP? It's 5 times 10 to the minus 20th. So now it all depends on the processing speed. Remember, the computers, the old computers were kilohertz, now megahertz, now it's gigahertz, right? The processing speed. I remember, what is the latest? 400? Something like this. That's the same for the brain, how fast you can change these bits. So now it all depends on what you use in terms of the biological material for this. It could be, a nanosecond probably is the fastest, so that'll be gigahertz frequency. It's like processing speed of the best computers. Gigahertz or nanoseconds are typical of protein conformational changes, up to microseconds. Most of the processes in the brain, actually, the action potentials last milliseconds. So it's a much slower processing time. So anyway, there is no single answer. It all depends on which unit and how fast it processes, but you have these limitations. 3 watts of power, no more, and 5 times 10 to the minus 20th of joule per bit change, and up to 10 to the 20th bits of encoded information in the brain. Okay, and this is the latest in this saga. I mentioned to you fractal organization. What really complicates these calculations tremendously, as you saw just a moment ago, is that there are different scales of time and space on which things happen. You have electronic scales, transitions very fast. You have protonic, fast but slower. Then you have protein transitions or enzymatic transitions, chemical reactions. And then you have physiological changes, which are the slowest. So it's like a structure within a structure within a structure. And this group of scientists from Japan, with Anirban being the lead author, proposed that actually there is a synchrony. These things are not independent of each other. The fast processes inform the slower processes, and the slower processes inform the slowest processes, and so on. So it's a scale within a scale within a scale. So I think that more or less is the scheme of things, how fast processes are not lost, but they are actually ignored, except for some important transitions. Okay, so I think I will be stopping here at this point. There's a bit more that I prepared, but that already is probably a lot. Maybe just in passing, I'll mention this. Anesthesia. Anesthesia is really the most interesting medical achievement of the last, maybe the most important achievement of medicine in the last 200 years. Because you cannot probably imagine going to the dentist, let alone to have an open heart surgery without anesthetics. Anesthesia enabled surgery and many procedures that otherwise would be unbearable to the patient, right? We still don't know, actually, how anesthetics work. Believe it or not, it was discovered 150 years ago, and we don't know how it works. The reason for this is that anesthetics have several actions, and they have also various chemical structures, and there is no rhyme or reason. There is no similarity between many of them, absolutely no similarity. They have four major modes of action. Analgesia, the blocking of the sensation of pain, which is why we take anesthetics in the first place. Hypnosis, so produces unconscious states in the brain. Amnesia prevents memory. You don't remember that there was something done to you when you were under anesthesia. And finally, paralysis. So the movement of more or less of the body is frozen during anesthesia. The amazing thing is that these are tiny molecules that switch off your consciousness, memory, and motor functions. And we don't know what happens, how it happens. There's only one kind of scientific law about anesthetics. It's called the Mayer-Overton relation. The potency of anesthetics measured in minimum alveolar concentration, or MAC, is inversely proportional to the water solubility or proportional to lipid solubility. In other words, if you have a very oil-soluble structure, it will be a very good anesthetic. Something which is solubilized in water is not a good anesthetic. These are just some of the most popular anesthetics in current use. Argon, xenon, single atoms, actual anesthetics, as well as molecules such as sevoflurane and halothane, which we studied. So I'll leave you with one thought. Molecules such as these can reversibly switch off consciousness, memory, things like that. And they do interact with tubulin. Anesthetics interact in many places with tubulin. They have low affinity, but they have many places where they bind. And so with my colleagues, we proposed that actually microtubules and tubulin in the brain are the sites of consciousness, because anesthetics, in addition to everything I said today, in addition to all the pathological changes in the brain, anesthesia is the most direct way of turning off consciousness and mental processes. And it all goes back to tubulin and microtubules inside the neurons, not receptors or synaptic connections between. This is the next level. I'm trying to focus your attention on the deepest level where things happen. So, yeah, we found many binding sites for tubulin, and some of that was experimentally demonstrated. So I just hope that this wasn't a waste of time. There's more questions than answers, as always. But you may have some questions to me. Thank you.
So based on the mechanism that we see that you hypothesize about the memory, so what do you think is going to happen when someone has, I know for example Alzheimer, people lost the memory, right? So there is something that is wrong with the mode of action of this camudin of the protein.
Kinase too. Yeah, so loss of memory, permanent loss of memory?
The degradation, I mean, what happened, yeah.
Okay, I think degradation is probably easier to explain because we do lose neurons with age. You know, the neurons never are born again, so to speak. They are not regrown. But there is gradual loss of neurons and probably, although I don't know, degradation inside neurons as well. Not just neurons having fewer and fewer functioning neurons, maybe fewer connections, but also inside. I would hypothesize that microtubules inside neurons are not as strong or maybe not as well connected.
Yeah, I mean the loss of memory that you can have if you have Alzheimer, for example. Okay, I mean, I think that you can get Alzheimer even when you are relatively young, right? I know that you can. There are some cases when you are like 50 or 45, you can get Alzheimer. So.
Oh yes.
And
yes, early onset.
Yeah.
But in terms of Alzheimer's, it's very specific. It's actually molecular defects. As I explained to you, there are two things happening: tauopathy, MAP-tau. The connection in microtubules are lost, and then there is beta amyloid plaque formation outside neurons, which, by the way, attracts zinc. It's actually growing. Zinc is the fuel for the plaque formation. At the same time, deprivation of neurons of zinc causing disassembly of microtubules. So that's, and I suspect that if you have, you know, by the way, this is an important question because Alzheimer's disease is very, and maybe Kim will echo this, you're an MD, I'm just a PhD, but Alzheimer is not easy to diagnose and quite often misdiagnosed. Dementia actually is quite often indistinguishable in terms of, you know, diagnostic, unless you do autopsy, post-mortem, then you don't know it was. You know, we don't have the tools to really diagnose precisely. But what I'm saying is that maybe degradation of memory, not catastrophic like Alzheimer's, but gradual degradation at the molecular level is not that different. By this I mean microtubule loss in neurons.
Yeah, I forget the details, but in the last week, there is some evidence that Alzheimer's type pathology can be reversed with microwave radiation.
Yes.
Have you seen those
yes,
reports?
Yes.
How does that
I think
fit into what you're talking about?
It's a great question. Also ultrasound stimulation, high frequency ultrasound and electromagnetic stimulation. I've seen some papers, including also magnetic fields, oscillating magnetic fields. So it's quite possible that you simply stimulate the regrowth of microtubules, or maybe break up some oligomers or clusters which are not properly assembled. You know, microtubules, as you saw in my presentation, again, apologies for the quality of the diagrams, but microtubules are very intricate. It's a cylinder with 13 filaments, 13 protofilaments, forming a very well-defined lattice. And pathologies may involve microtubules which are not properly formed. And therefore, if you shake it up with electromagnetic or ultrasound, you may actually break up the faulty microtubules and allow the cell to reassemble properly. That will be my immediate reaction to, you know.
Yeah, well, it's a very hopeful development, I think, because it seems to have this positive effect without, you know, damaging surrounding cells. So.
I seem to recall that in the last few years there were like 20 clinical trials for Alzheimer's medication, new drugs, all failed. There's
yes. It's been pretty dismal up until now. Okay, well, thank you very much for an outstanding teaching session, 2 outstanding sessions. The first time we ever succeeded in doing it twice, so it's a landmark day.